Hello everyone, we are a small group discussion B10 from the Bachelor of Medicine and Medical Doctor Study Program of Udayana University 2022. Today we will present you our student project presentation and the title of PCR application for diagnostic purpose. Now let's dig into the topic.
Hello, my name is Komang Bunga Sedani. With the last three-digit number is 241. I want to tell you the background about our topic that we brought today. The first one is the rapid development of science and technology is the same line with the motto of medical student, long life learner. We certainly realize, if we do a flashback, the development of science and technology that has existed from time to time has had a huge influence to the civilization in the world. As a medical student, we have a motto that is long life learner, which means that until the end of our career, we will always be required to be literate with the new updates from science and technology to what it has developed, especially in the health sector, to reach the ultimate goal of providing the best service to our patient. Second one, the PCR technique has a very important role as a technique that is widely used in case study method in the healthcare. The synthesis and amplification technique of the DNA, where we can do the multiplication of DNA segment 1,000 or even million more times with only the help of a tool called thermocycler within a few hours. In this PCR technique, to be successful, it will undergo 3 cycles, that is denaturation, annealing, and extension. This technique is certainly very useful in the health sector. Through this technique, we can perform analysis, detection, identification, and diagnosis of a matter related to the information carried by a living being. For the example, it is organism, or no matter what it is. Therefore, this project will mainly review the application of the PCR technique for diagnosis purpose in the sector of the health science.
Hello everyone, let me introduce myself. My name is Hana Korestanadin and my 3 last student ID number is 236. And today, I'm going to tell you guys about the purpose of PCR application. So, understanding the PCR application for diagnostic purposes, we create student project entitled PCR application for diagnostic purpose with the following purpose. First, understanding the definition of PCR. Second, understanding the component in PCR. Third, knowing the advantage of PCR. Fourth, knowing the disadvantage of PCR. Fifth, knowing the type of PCR. And lastly, knowing the procedure of PCR. Thank you.
Hello, let me introduce myself. My name is Komang Utama and my last 3-digit study number is 221. And then now I will give benefit about our topic. The PCR application for diagnostic purpose tends to increase effectiveness in determining the presence of the coronavirus in individuals. PCR is made to help its healthcare providers to identify the presence of the virus. It helps assist to identification process because PCR use DNA from individuals that were tested in labs. Okay, that's all about our benefit. Thank you.
Hello everyone, my name is Rafa Diardra Fajri and my last 3 digit of my serial number is 244. Definition of polymerase chain reaction. Polymerase chain reaction, also known as molecular phototyping, is a technique for amplifying small target segments of DNA to generate millions of copies of a particular gene fragment. PCR cycle has 3 important main steps. First is denaturation, the second one is targeting or annealing of specific primers, and the final one is extension. This PCR process produces 2 fold copies of 1 into 2 into 4 into 8 into 16 into 32 and so on. A single PCR process can be completed in 30 to 35 cycles and take 2 hours to amplify and generate usable quantities of DNA fragments. That's all from me. Continued by my friend.
Before I continue the presentation, allow me to introduce myself. My name is Gustia Erika Nananita with last 3 digit of student number 233. Here I will explain about the components needed for PCR. The first one, there's the DNA template. The DNA template will be used as a sample in amplification of DNA sequence by PCR. The DNA template, which contains target sequences, can be added to PCR in the form of a single strand or a double strand. The second one, there's primers, which are single-stranded DNAs that are usually around 10 to 30 nucleotides in length and must be complementary with both ends of the sequence that will be amplified. The third one, there's nucleotides. There are 4 deoxyribonucleotide triphosphates, which include adenine, guanine, cytosine, and thymine, which are needed to provide building blocks for DNA replication. Each complementary base will be added to the new growing DNA strand by DNA polymerase and are added according to the original strand sequence. The fourth one, there is DNA polymerase, which is an essential enzyme needed for DNA replication. Since extreme heat is involved in PCR, a specific type of DNA polymerase is needed, which is Taq polymerase. Taq polymerase is able to withstand extreme temperatures of around 100 degrees Celsius and does not easily denature in extreme temperature. The last one is buffer, which is also needed to maintain a stable pH level and provide the magnesium ion cofactor needed for DNA polymerase to function properly. That's all from me. The next part of the presentation will be continued by my friend. Thank you.
Hello everyone, my name is Igusti Ai Prasapita Hasikirana, and the last three digit of my student ID number is 212. Deoxyribonucleic acid, or DNA, is continuously multiplied through repeated cycles of denaturation, amplification, and replication in PCR. Afterwards, specific DNA primers are bound to indicate the presence of the target cells or microorganism. The PCR method has been used throughout molecular biology for cloning and sequencing genes in order to detect mutations that may exist within them. This method is extremely effective for detecting microorganisms, requiring only a small sample and taking only 4 to 8 hours to complete, which is 3 times faster than the culture methods. There have been many studies showing PCR can be as good as, if not better than, traditional staining and cultivating method. Given culture and stains for both bacteria and fungi, polymerase chain reaction demonstrated high positivity and sensitivity. Zhou et al. Reported the positive detection rate of PCR as 83% and 84% for bacterial and fungal keratitis, respectively. For fungal keratitis, this rate was greater than the positive detection rate for culture, which is 35%, and stain, in the number of 65%. Overall, studies have reported that PCR has a higher sensitivity compared to the culture and staining methods, and also has a high possibility to detect less common organisms, such as viruses, which can be seen in its use to detect the existence of COVID-19 in these days.
Hello everyone, so let me reintroduce myself. So my name is Hannah Claristanadin and my 3 last student ID number is 236. And today I'm going to tell you guys about disadvantage of PCR. The first one is PCR potentially lower specificity compared to culture and staining. Second, need to narrow list of causative agent to use specific primers. Third, possibility of amplifying normal flora from corneal scraping. Fourth, becomes less cost-effective when performed with a multi-organism PCR approach. And lastly, supply cost, machinery fees, and training expenses. That's it from me. Thank you.
Hello, my name is Jinariska Pramesti with the last 3 digits student number 208. Now I'm going to explain about the type of PCR. So PCR is divided to 6 types: quantitative, qualitative, conventional, multiplex, nested, semi-nested, and real-time PCR. Quantitative PCR, it is known also as real-time PCR that provides information about the amount of DNA in the sample. Qualitative PCR used to identifying a particular DNA fragment, and it can tell if a person has been reinfected with a different but related pathogen. Qualitative PCR is quick, easy, and cheap procedure. Conventional PCR, aluminum block PCR tubes, DNA polymerase buffer, target DNA, and primers are all that are required for the PCR process. The entire procedure is performed in 35 to 40 minutes and observed using the gel electrophoresis method. Prime star HS gates can be used to perform the PCR procedure. Multiplex PCR technique is employed to find bacterial or viral and other infectious agents. In addition to buffer, Taq polymerase additive is used in multiplex PCR, which lessen rivalry between applicants. Nested semi-nested PCR, the purpose of nested PCR is to lessen product contamination brought on by the amplification of anticipated primer binding sites. The last one is real-time PCR. Is a nuclear-derived technique for determining whether any pathogen, including a virus, contains a particular genetic material. The most used laboratory technique for detecting the COVID-19 virus is real-time PCR. Although many nations have employed real-time PCR to diagnose other illnesses, including the Ebola and Zika viruses, many still want assistance in adapting this technique for the COVID-19 virus, along with expanding their national testing capabilities. That's all from me. Thank you.
Okay, before I start, my name is Nimade Aprilia Dwi Sisyanti with 3 last digit of student number 229. In this presentation, I will explain about the procedure of PCR. PCR have 3 steps that repeat iteratively. There is denaturation, annealing, and also extension. Temperature cycling is a technique used in PCR to start and stop bursts of enzyme-catalyzed DNA synthesis. This procedure is carried out in a thermocycler, a programmable machine that regulates the temperature and timing of each cycle step. The first one is denaturation. The temperature at which double-stranded DNA takes place. Denature depends in part on the amount of G plus C in each strand. Denaturation in Taq DNA polymerase catalyzed PCRs take place around 94 to 95 degrees Celsius. Denaturation is sometimes carried out for 5 minutes during the first cycle of PCR to maximize the likelihood that lengthy molecules of template DNA will be thoroughly denatured. Okay, the next is primer annealing for DNA template. The annealing steps temperature or TA is crucial. The yield of amplified DNA is very low when the annealing temperature is excessively high because the oligonucleotide primers only partially or never anneal to the template. By subjecting a single PCR to a sequential series of annealing temperatures in subsequent cycles of reaction, optimization is achieved rather than scanning a variety of annealing conditions in separate PCRs. And the last one is extension. The thermostable polymerase catalyzed DNA synthesis at temperature that is ideal for oligonucleotide primer extension. Extension from one primer move past to the sequence that is complementary to the other primer's binding site in first 2 cycles. And extension is typically carried out for 1 minute every 1000 BP of product. That's all about the procedure. Next material will be continued with my friend. Thank you.
Hello, my name is Melissa Cynthia Dewi with the last 3-digit student number 223. Today, I will tell you about the principle of PCR. As we all know, the polymerase chain reaction, or PCR, was discovered by Mullis in 1983, and it was carried out by in vitro replication of specific DNA sequences with the help of enzymes. In PCR, the target DNA will be copied by thermostable polymerase enzymes with the help of nucleotide and primers. This process will later produce thousands of billions of specific true DNA extracts. Basically, PCR occurs through several stages, including denaturation, annealing, and elongation. Initially, true DNA sequences called primers for binding the target DNA are created. Primers containing nucleic acid and will find the target amplification to increase. The DNA template containing the target was added to the tube containing the primer, deoxyribonucleotide, and DNA polymerase enzyme, and then fed into the PCR machine. In the PCR machine, the DNA template mixture is previously and DNA polymerase enzyme, and then fed into the PCR machine. In the PCR machine, the DNA template mixture is previously heated to separate the double-stranded DNA template into a single strand, and then it cooled so that the primers melt and bind to the DNA template. After that, the temperature will be raised to 72 degrees Celsius to optimize DNA polymerase so that new DNA strands are synthesized. This new DNA segments serves as a template in the next cycle to amplify the next DNA target.
I will continue this presentation about the principle of the PCR. Before that, let me introduce myself. My name is Tabita Eterina Yunia Brusumbiring with last 3 digits of student number 232. Next in this section is the denaturation. Denaturation is the separation of 2 DNA strands which can be obtained by increasing the temperature. At this temperature, the matrix DNA that serves as the matrix during replication will be denatured and the double-stranded DNA will be denatured to single-stranded DNA. Second, hybridization. Hybridization is carried out at a temperature which is called the primary hybridization temperature. A decrease in the temperature allows hydrogen bonds to reform as well as complementary strands for hybridization. The higher temperature and the more selective hybridization, the more specific the hybridization will be. Third, elongation. Elongation is a complementary strand synthesis at an elongation temperature. At this temperature, tag polymerase binds to prime single-stranded DNA and use the deoxynucleotide 3-phosphate present in the reaction mixture to catalyze replication. Therefore, the region of template DNA downstream of the primer is selectively synthesized. Fourth, primers. To achieve selective amplification of nucleotide sequence from DNA extracts by PCR, it is essential to have at least one pair of oligonucleotides. These oligonucleotides, which serve as primers for replication, must be chemically synthesized and have the highest possible complementary to both ends of sequence to be amplified. And last, tag polymerase. DNA polymerase that is used has been isolated or cloned from the Thermus aquaticus. This polymerase has an ability to survive temperature around 100 degrees Celsius, which are typically sufficient to denature the majority of protein.
Let me introduce myself first. My name is Ida Prartini Surya with the last 3-digit student number 230. Today, I would like to present about the applications of PCR to the diagnosis of genetic disease. But first, we should know what is the PCR. Polymerase chain reactions, or PCR, is a technology in the form of tests to rapidly develop and amplify copies of certain DNA segments. PCR is an indispensable tool in applications of molecular biology and beneficial for diagnostic medicine. Because many scientists believe that all diseases have genetic components, and because gene affects almost all traits that are formed in our bodies. Genetic diseases generally are caused by mutation that begin from simple chains in the base sequence of DNA double helix to large DNA sequence chains and even chains in the entire chromosomes. PCR is used to detect whether a segment of related DNA is the same as the DNA of a pathogen. In the context of genetic disease diagnosis, PCR is used as a tool for gene checking. Gene analysis as a PCR product was carried out by electrophoresis. It is to determine the size of the gene, which led directly to the diagnosis. That's all. Thank you.
Hello everyone, my name is Farel Armando Damanik, and my last 3-digit of student number is 243. And I'm going to continue the explanation of what my friend have told, which is about the diagnosis of the genetic disease with the PCR. So in some cases, such as inversion and point mutation, the difference in size between the DNA of healthy and sick people is not visible, or in other words, the difference is zero. 3 approaches can be chosen, one of which is restriction fragment length polymorphism, or FLFP. In addition to the PCR, for PCR FLFP, ligation with restriction enzyme is needed, and the obtained restriction fragment, or FR, are separated based on their lengths by the aid of agarose gel electrophoresis. The RFLP can detect inversions such as point mutation. It involves a restriction enzyme capable of hydrolyzing the PCR product at the sequence which sets the mutation. This approach is only possible if a restriction site is indeed present on this sequence, is indeed where it is the mutated allele or the wild-type allele. The restriction enzyme thus hydrolyzes either the PCR product derived from healthy DNA or that which is derived from the diseased DNA. From these PCR products, 1 or 2 DNA fragments are thus obtained, which are then revealed by electrophoresis, which method detection is, like the FLLP, adapted to inversion and point mutation. PCR product consisting of sample DNA and reference DNA, which are each contained from the DNA of the patient and the DNA of the normal healthy person, are going to be mixed. It is then going to be denatured by increasing the temperature and then rehybridized.
Let me introduce myself. First, my name is Komang Tlangkas Atnya Sujatyana with the last 3 digits student number 224. And now I will give a conclusion about our topic. It has been an obligation for us as medical students and also for all sides that are working in the health sector to be responsible for continuing to follow and develop medical science. It's because the development of medical science are needed to facilitate and create effective treatment. One of the developments of this science is the PCR technique. PCR is the one of the result of the development of science and technology. PCR allows us to duplicate DNA segment to produce millions of times even more with the help of a device called a thermokiller. In just a few hours. For better result, many types of PCR process can be used with slick modification include beam multiplex PCR, RT-PCR, nested PCR, inverse PCR, colony PCR, asymmetric PCR, helicase PCR, and ligation-mediated PCR. The material needs to perform PCR are DNA templates, primers, nucleotides, DNA polymer, and buffer. PCR also has advantages and disadvantages. The advantages of PCR are high sensitivity compared to culture and staining, rapidity in 4 hours, increased ability to detect less common organisms such as virus. On the other hand, the last one is the advice for the research in the future. Based on what we have already discussed above, the need for the development of molecular medicine is very important to improve the quality of prevention, diagnosis, and therapy. Applications that have been proven to work effectively to improve the accuracy of this treatment must continue to be developed in the future. This can be done by fixing PCR weakness and turning them into the strengths of PCR. Therefore, hopefully, the research in the future of the PCR method could see how the PCR can be improved again to perfect this technique. Because the PCR method is very sensitive, contaminants and annealing errors in DNA primers are factors that are difficult to avoid. Thank you.